It's the day we've all been waiting for. Today, the team gets to find out whether their vaccine candidates, Q or Z, provide a protective immune response in mice. In other words, is Q or Z the breakthrough we need against Chappare mammarenavirus? Welcome to Day 0 to Breakthrough. Let's find out. A few days ago, the team started processing their samples from the animal study. The team isolated antibodies and also a type of immune cell called B cells, which are important in antibody production. I followed Nick as he set up something called a pseudovirus assay, a test which will give a good indication of whether Q or Z provides a protective immune response against the Chappare mammarenavirus viral fusion protein. Since the Chappare mammarenavirus is such a dangerous pathogen, the team uses a pseudo or fake version of the virus, which, when used with proper protective measures, is quite safe. What they did was take a different kind of virus, called a lentivirus, which they modified so it can't replicate, and told it to express the Chappare mammarenavirus viral fusion protein. This modified lentivirus also contains a gene that causes infected cells to fluoresce. Nick has mixed the pseudovirus with antibodies from the animal study and allowed them to incubate. If there are antibodies in the mix that can stop the pseudovirus from binding and infecting cells, then we will see less fluorescence than if no antibodies were present. Nick added the mix of pseudovirus and antibodies to a plate containing cultured cells, which will be allowed to incubate over the weekend, and we'll get the result on Monday. I took the opportunity to check in with Keith and see what he was thinking coming into the most important readout of the pressure test so far. He highlighted a key challenge that comes with this kind of pandemic response.
Part of the difficulty is we needed to build the assay at the same time as doing the mouse immunisations. And to do that, we don't have a positive control. We don't know if the assay works. So we're really going into this blind. So if it doesn't work, we don't know if it's because the assay is no good or if we don't have an immune response. So if it succeeds, it's going to be fantastic. If we fail, we're going to have to do a lot more investigation to figure out why that is. But fingers crossed for a good result on Monday.
Now for the moment of truth. Did the pseudovirus assay indicate that Q or Z could provide a neutralizing immune response against Chappare mammarenavirus? The answer is no. Emotions on the team range from disappointment to continued, cautious optimism. I asked Keith what this result meant for the team moving forward.
I think this is research, so nothing's going to go perfectly, and that's why we're doing the pressure test. We're trying to work out what works smoothly, what goes wrong, how we can do things quicker and faster and more effectively next time around. So it's not at all surprising to me. I've had a career in research for the last 20 years. There's been plenty of times where things haven't gone to plan.
I'm continually impressed by Keith's optimism in the face of setbacks and determination to keep moving forward no matter what.
This is a test, right? So if we don't get to the end point, that's okay. There are things within this trial that have worked extremely well, and there are things that are difficult and have failed this time around. To me, that's the whole point of why we're testing this and why we need to regularly run these pressure tests. So the stage we'll go through at the end of this is to look back and figure out, this part didn't work. Why didn't it work? How can we be more confident we get it to work next time around?
So, what next? Are the team's hopes of breakthrough over? Certainly not. The team have plenty of other experiments they want to perform to help them understand their vaccine candidates better, including why they didn't perform in the pseudovirus assay as expected. They want to do an ELISA, which will help them see if any antibodies bind to the viral fusion protein at all. And they're also going to engage in some exciting pursuits such as B-cell discovery and phage display. A key part of the pressure test is making sure the team can scale up production of their vaccine candidates for use in a human clinical trial. Even though the vaccine candidates aren't performing as expected, the team is moving forward with this part of the process because it's important to test under pressure. This means that next week we'll be visiting the National Biologics Facility, who are busy producing a large-scale batch of our vaccine candidates. Stay tuned, we're still on track for Day 0 to Breakthrough.